Hello students, in this video we are going to talk about gel electrophoresis. Now what is the meaning of electrophoresis? This gel electrophoresis method was developed by A. Tiselius. A. Tiselius. Now what is gel electrophoresis? Gel electrophoresis में what is done is, it is the separation of the molecules of, so what is gel electrophoresis? Separation of molecules like DNA or RNA or protein based upon their size or length under the influence of electric field. Under the influence of electric field is called as gel electrophoresis. Now, literally what is the meaning of electrophoresis? So, electro का मतलब है electric field, फोरेसिस का मतलब है to run. So the DNA fragments या RNA fragments run on the gel based upon their size or length. Okay, they are moving from one electrode to another. That means they are moving from cathode towards anode because DNA fragments are negatively charged. Now, this gel electrophoresis is carried out in a gel, as the name suggests, and the gel is called as agarose gel. Carried out on agarose gel. What is agarose gel? What is agarose? Agarose is a polysaccharide which is obtained from seaweeds. And this you have done in the chapter Plant Kingdom, Gelidium or Gracilaria. From those seaweeds we get this polysaccharide called as agarose, on which the DNA fragments are run under the influence of electric field. Okay, so now I will quickly explain you the procedure of gel electrophoresis in short. So in gel electrophoresis, they use a tray on which there is agarose gel. So they use a tray of agarose gel. On this, now how this agarose gel look? You must have seen a wax का tray which we use in the lab. एक छोटा सा tray होता है जिसके ऊपर wax होता है. Okay, wax tray. Like that, we also get agarose. We can also set agarose gel in the tray. अभी इस gel में क्या करते हैं? They literally make some wells, very small wells, like this. So these are the wells in which, now what they do is, this agarose gel is connected to a power supply. Is connected to a power supply, positive, negative, whatever it is. Now, in this, they will add the sample of DNA. क्या ऐड करेंगे वो? DNA का सैंपल. Now, all the DNA fragments, यहाँ पे क्या किया है? पहले the DNA has been extracted DNA is fragmented by restriction enzymes, and these fragments are poured in the gel, poured in the wells यहाँ पे. Fine. Now the power supply is on. Power supply is on. Now these fragments will run on the gel. How they will run on the gel? The smaller the DNA fragment, it will run more fast. So these are the DNA fragments. The DNA fragments will run on the agarose gel. DNA fragments will run on the gel based upon their size, because the pores are created in the agarose gel through the pores of the gel. Okay, so smaller, see, agarose gel is porous, so DNA fragments, if it is very larger, it is going to remain behind. If it is smaller, then it is going to come fast. Now these are the sizes of DNA fragments in kilobases. So the small fragment will be 100 kilobases, then 200 kilobases, then 300 kilobases, then 400 kilobases, right? So जो DNA fragment सबसे बड़ा होगा, it will be at the other end, मतलब it will be at the starting point, और जो DNA fragment सबसे small होगा, it will be at the other end. Fine. So this is how the DNA fragments will run on this agarose gel. Based upon its size, the charge on the DNA will be the same. It is based on the charge to mass ratio that the DNA fragments are running on the gel. Fine. Now, तो ये तो हो गई अपनी DNA fragments the gel electrophoresis की procedure. Now the question is very basic question कि why do we have to perform this gel electrophoresis? करते क्यों हैं? What is the requirement? Electrophoresis से हमें क्या मिलेंगे? DNA fragments मिलेंगे, right? Because we want DR insert जिसको हमें vector के साथ join करना है. We want to use it for our DNA technology. So we need to separate the DNA fragments from the complete extract, right? So cell से हमने जो extract निकाला था DNA का, उसको cut किए बगैर we are not able to use them. So you have to first cut them into pieces and then use the piece which you want for ligation with the vector. Okay. So now what we do is, after completing this gel electrophoresis, these DNA fragments are still not visible. They will be made visible by staining. So the DNA bands are made visible by staining with a stain, or we can say a dye or a color, which is called as ethidium bromide. Ethidium bromide. So a question is asked on this ethidium bromide regarding the DNA fragments after gel electrophoresis are stained with dash stain. So it is ethidium bromide. It is written as ETBr. So DNA fragments are stained with ethidium bromide and they get an orange color. So you observe orange colored bands. Orange colored bands. But again, this is also not sufficient. Staining, after staining with ethidium bromide, what is done is these fragments are exposed to UV light. After exposure to UV light, these fragments you will observe as orange colored bands. Alright, so this is very important कि DNA fragments जेल पे आपको नहीं दिखेंगे, उसके ऊपर आपको स्टेन डालना है एथिडियम ब्रोमाइड. स्टेन डालने के बाद आपको उसे अल्ट्रावायलेट लाइट में देखना है, देन यू विल ऑब्जर्व दिस ऑरेंज कलर्ड बैंड्स. नाउ अभी नेक्स्ट पॉइंट ये है कि हम इन फ्रैगमेंट्स को निकाले कैसे एंड हाउ व्हाट इज द प्रोसीजर. सो दैट वर्ड यूज्ड टू extract the DNA fragments from the gel is called as elution. So what is elution? Elution का मतलब है to remove the DNA bands from the gel, or to separate, remove separate the DNA bands from the gel. This word is called as elution. So we can also expect one question on this elution. What is elution? Separation of DNA bands from the gel is called as elution. Now after elution, what do you do? After elution, we can use these DNA fragments either for constructing and our DNA. मतलब these DNA fragments can be of vector, it can be of insert that you want, and you're using them to construct a new recombinant DNA. Alright, so this is very important process of gel electrophoresis. I hope this process must be clear to all of you. This process is generally asked in your exam. What is the gel used? सबसे important question ये है कि fragments कैसे run होंगे gel पे. जो छोटे fragments है, they will go to the rare end because उनका size छोटा है. Smaller the size, the longer they run. So remember this tagline: smaller the size, the longer they will run. If they are larger in size, they will not run very fast on the gel because the gel is porous and their movement will depend only on the size because their charge is always the same. Second important thing you will remember is ethidium bromide. You stain DNA fragments with ethidium bromide, they appear orange in color, but you have to observe them under ultraviolet light. Okay. Elution is a technique by which you remove the DNA fragments out of the gel for your construction of recombinant DNA technology. So this was gel electrophoresis.